So let's get into business. Today's webinar, as you've seen on the title and as on the description, will be about how we've applied the technology that we've developed at Pixelgen Technologies to study CAR T cells. And I plan to go through a little bit of background into how this technology works and what kind of output we provide. We have a lot of more information on the website. There were recently a webinar really going into depth about the technology and the workflow, etc., that you can also access on our website and on our YouTube channel as well. So we will keep that part a little bit brief and then really focus on the results on CAR T cells. So what we offer then is basically a method for measuring protein interactomics. And what is really novel is that we can do this in single cells. And we do this by creating so-called proximity networks. And just to tell you super shortly about the company, which is called Pixelgen Technologies, we're a Swedish biotech company. We were founded in 2020. We have very recently released a new product that can do this protein interactomics. And we're now arranging these webinars to kind of showcase how you can use this assay. So let's get into some background. Why do we want to study single-cell protein interactomics at all? So I think most of us are kind of aware now that cell function is really dependent on membrane proteins. And although for kind of discovery and high dimensional studies, RNA is still commonly used, it's because we have a good technology, single-cell RNA sequencing for that. But good single-cell proteomics methods are being developed very rapidly now, since we're really realizing that proteins are key to cellular function. However, typically it's not really enough to just know if a protein is there or not, because the way that the cell is regulated by these proteins is typically by these proteins reorganizing. So, So, for example, when a cell is getting engaged by ligands, these will bind to the receptors and the receptors can reorganize and thereby induce signaling into the cell. There's also formation of multi-protein complexes, clustering, all kinds of reorganization that regulates the function. Cellular function is most often dysregulated in disease in one way or another, and the underlying kind of altered protein organization that leads to this dysregulation in disease is really a very untapped source of biomarkers. So this kind of dimension has not been explored at a high resolution, high multiplexing yet. And there are really potential to find new biomarkers and also druggable targets by starting to look at this new dimension of protein organization. So I'm sorry, I have some technical issues. Okay, so just some examples of cases when protein organization actually matters in immune cells. So depending on what proteins are interacting on the cell surface, you can have very different responses to ligand binding, or the ligand might not even be able to bind depending on what interactions are happening on the cell surface. So for example, on tumor cells and antigen-presenting cells, you can have cis interaction between checkpoint molecules and other receptors, and this can really influence the response to checkpoint blockade. You can also have kind of spontaneous clustering of receptors, as is seen, for example, in CAR cells or in B cancer cells, where the B cell receptor is spontaneously clustering. And this is inducing signaling to the cell, which leads to enhanced proliferation in the case of cancer, or to actually tonic signaling and exhaustion in CAR T cells. And this is very much regulated by the organization of the receptor. And if it's forming clusters, it can start to accumulate signal and start to signal, even though it's not really binding its ligand. Of course, maybe the most well-known example of where we have a lot of protein reorganization is during cell-cell communication. So for example, during synapse formation, you will have really big, large-scale changes in the protein organization. And this really enables the cell to respond in a directed manner, to really only kind of release its response towards a cell that it's in close contact with. And this is, of course, particularly important for example, cytotoxic immune cells, where you want to kill a tumor cell or a virus-infected cell, but you really need to make sure to not damage any surrounding tissue, for example. So kind of after explaining here why it's so important to look at protein interatomics, how can we do this? And what kind of technology do we offer to do these kind of studies? So today I will present the PixelGen Proxiome kit. And this method makes it possible to do protein interactomics by proximity networks, as I mentioned in the intro slide. And this is actually a sample prep kit I will show you in the coming slides that makes it possible to generate these kind of 3D layouts of individual cells and individual proteins on these cell surfaces. So for example, here you can see around 50,000 unique molecules and where they are located on an individual cell. So it's a really powerful technology for measuring protein interactomics. So just shortly how it works. Basically, this is a method for cells in solution. So it can either be single cells in solution or, for example, cell complexes in solution. And then what you do is that you apply this kit, and this is actually a sample prep kit. So it's something that you do during the sample prep. You go through this protocol with particular reactions. And as I mentioned, there are way more information about this on the website. And basically what you will end up with is a sequencing ready library. And this library can be sequenced in bulk on any Illumina platform. And following this sequencing, you perform data analysis, and this enables you to regenerate the 3D surface map of every single cell. So here you can see an individual cell, for example, or a cell-cell complex where you have two different cells that are interacting with each other. So as I mentioned, this is done by sequencing, so a bit untraditional if we're used to looking at the protein organization. I think most people are immediately thinking about microscopy when you do that association. But here we have developed a technology for doing this by sequencing. And the really powerful thing with sequencing is that you are not limited by the multiplexing. So it's possible to do so many more markers than you can do with microscopy. So how can we actually get the protein localization using sequencing? Let's go through the protocol just briefly. So as I mentioned, you start by using cells in solution. And the first step of the protocol is to fix the cells. And the reason why we want to do this is to immobilize the cell surface proteome to make sure that no proteins can move during this protocol. In the next step, we add a 155-plex antibody panel. And in this panel, every single antibody has a unique DNA barcode. And this enables us to detect every single antibody actually within our cells. So we stain the cells in solution with these 155 markers, and they will bind to their respective targets. After this, we do rolling circle amplification of these unique sequences. And the reason why we do this is to make them a little bit longer. So after we have extended them, we can start to connect these unique sequences that are close to each other in proximity. So we add a very short linker sequence. They will be able to connect these products that are very close to each other in space. And these are actually these linker sequences that we sequence in the end. So every single linker sequence will, after the protocol, contain information about which two proteins it connected. And all of these small sequences are sequenced in bulk. And then, as I mentioned, computationally, we actually rebuild the entire network of every single cell. So since we know exactly which proteins are connected to which proteins, we can rebuild the entire network of every single cell. So if you input 1,000 cells to your reaction, you will get 3D single cell data for 1,000 cells in the end of the protocol. So when it comes to the data that you get out of one of these experiments, you can generally divide it into 3 different parts. So since this is a single cell method, you can still look at single cell protein abundance. So how much of a particular protein is expressed. And this is measured on a single cell level for every single marker on every single cell. So you really get a lot of information since you have 155 markers in this panel. But what is really novel and what you cannot do with, for example, CiteSeq that also measure protein abundance by sequencing is to get spatial information about where these proteins are located. So one feature that you can measure is, for example, protein clustering. And basically what we then want to look at is if a protein is evenly dispersed on the cell surface or if it's forming some kind of clusters and co-localizes with itself. And in order to measure this more efficiently, we have also developed a clustering score that actually does an assessment of this at the single cell level. The third output that you get from one of these experiments is co-localization. And the co-localization is a pairwise comparison. So basically you look at two different proteins and you see, are these two proteins localized in relation to each other in a random fashion? So not co-localizing, not really segregating from each other. If this is the case, our co-localization score will give you a 0 score. On the other hand, if these two proteins are actually co-localizing more than you expect by random chance, you will get a positive score. And if these proteins are segregating from each other, so really not co-localizing together, you will have a negative score. And this kind of assessment is done pairwise for all of the 155 markers in the panel. So you will be able to, of course, look at single protein complexes, just constituting of two different proteins. But since you do this very high multiplex comparison, you will also be able to look at more complex structures like multi-protein domains, how many different proteins are organized in relationship to each other. And this is really, really difficult to do with microscopy since you're very much limited in the number of fluorophores you can use at the same time. So that was it for the background on the technology. Please, if you have any questions about the technology, we can take them at the end of the presentation, and you also have more information on the website. And now I think we should dig into the application note and the CAR T cell example that I wanted to talk about today. So the application note is called Uncovering the CD19 CAR T cell protein interactome using the Proximity Network Assay. And this was posted, I think, last week or a few weeks ago on our website. You can also reach it here using this QR code. So basically, this application note is focused on phenotyping of CAR T cells. And in particular, we are showing how you can investigate receptor organization, really seeing how the CAR receptor is organized, if it's clustered or if it's evenly distributed, etc. We also focus a lot about understanding what are the CAR receptor interaction partners. So what other proteins does the CD19 CAR receptor co-localize to? Of course, we measure protein abundance in these CAR T cells, and we also do this during stimulation, as you will see, so we can look at differential protein abundance. And finally, we're also looking at how you can use this technology to look at functional responses in these cells. So why do we actually want to use this technology for looking at CAR T cells? Well, the membrane organization is really key in all immune cells, all cells in the body in general, and of course then also in CAR T cells. So let's go into a few examples where the organization of the CAR can actually influence the function of these cells. So here's just a short schematic of how the CAR receptor looks like that I borrowed from a review paper. So basically, a CAR receptor consists of an antigen recognizing region. And this can be, for example, against a tumor antigen. There are also CAR developed against autoimmune targets, etc. currently. So first, you connect this tumor recognition site to a transmembrane domain. And this transmembrane domain also needs to contain some kind of signaling unit in order to make this a signaling receptor. And that signaling unit was originally taken from part of the TCR complex that we know induces the correct response in T cells, that it can induce cytotoxism and interferon production, etc. So this signaling domain, CD28 in that case, and the CD3 epsilon was connected to the CAR receptor. And this allows you to create a receptor that can recognize a tumor antigen and that can signal in a kind of cell-intrinsic manner. So, for example, here we have a CAR T cell that is expressing this CAR receptor, and it can recognize a tumor antigen and thereby induce cytolysis of this tumor cell. What is really interesting and kind of the first note here is that depending on how you design this CAR receptor and exactly which elements you put in this receptor, it can really impact the organization and the cell signaling. So, for example, different transmembrane domain can cause the receptor to aggregate or not, and this can influence tonic signaling, for example. It's also common now to try to develop CAR receptors that are actually less sensitive or that are inducing less signaling. So although the cells are maybe less functional short term, they manage to keep their functionality and not get exhausted by doing these alterations. However, it's not really completely understood, depending on how you change this membrane domain, what kind of changes you get in signaling and how this actually is affected by organization. There's a lot to learn here. As I mentioned shortly, receptor clustering also really affects the CAR T cell function. So with CD19 and other hematological targets, for example, BCMA, there's very little self-aggregation of the CAR receptor, and this has been shown before. However, with certain targets certain antigens that target solid tumors, so for example, GD2, it's been reported that there's high degrees of tonic signaling, and this is actually related to spontaneous clustering of the CAR receptor. And depending on if you change parts of the transmembrane domain, you can actually limit this tonic signaling. So here, of course, it's very interesting to understand what different receptors are self-clustering and how does this regulate tonic signaling. There has also been some reports where some studies where they have actually induced increased aggregation of the CAR receptor and actually caused better function of the cells. It's really not well understood what is kind of causing the dysfunction and how this relates to the protein aggregation. When it comes to reorganization of proteins, another feature that's really well kind of appreciated to play importance is trogocytosis. So for trogocytosis is a process where cells transfer membrane between different cells. So for example, what can happen in this process where you have a T cell interacting with a tumor cell is that the T cell can actually pull some of the membrane from the tumor cell, thereby acquiring some of the receptors from the tumor cell. This has been shown to, for example, lead to antigen escape. So the CAR T cell is pulling antigens from the tumor cells, and eventually the tumor cells don't really have enough antigens to trigger the T cell. There are other reports where it's been shown that the CAR T cell can actually share its CAR receptor, and you can thereby kind of make CAR T cells from cells that were actually not transfected with the CAR receptor originally. But the process of trogocytosis can really impact the kind of long-term effect of your CAR T cells. It's also a very important feature to consider. Okay, so let's go into the experiment that we did for this particular application note. So in this particular project, we were interested in looking at the CD19 CAR. And we acquired anti-CD19 CAR T cells with the FMC63 domain from a commercial source, so from ProMab. And then we used our 155-plex panel to detect 155 markers on these CAR T cells. And we also used FMC63 directed barcoded antibody to be able to actually detect the CAR receptor itself. So here you can see a high-dimensional clustering of our sample, our CAR T cell sample. And you can see that this sample contained both CD4 cells and CD8 cells. This particular sample had more CD8s than CD4s. And it was about 40% of the cells that were CAR positive, both in the CD8 and the CD4 population. What is really neat here is, of course, that every single cell that we're plotting here, you can actually look at it in 3D. So here I'm displaying you one CD8 CAR T cell, where we're plotting the CD19 CAR receptor in green. And you can see that the receptor is really evenly distributed all over the cell surface and at quite high levels. And this is very much expected since we know that the CD19 CAR is quite homogeneously expressed on the cell surface. So next we went in and we wanted to characterize how does the CAR T cell look like? What are the protein domains? How do the different proteins organize? So what I'm showing you here is a colocalization heat map, where every single square here represents a colocalization score for a protein pair. So for example, if we look at, we can take a square here. This is the colocalization of CD82 to HLA-DQ. And if it is a positive score, so if they are showing positive colocalization, it's red. And if it's close to 0, it's white. And if the proteins are actually segregating from each other, it's a blue color, as you can see. So here you can find all the different protein complexes that we expect. So for example, HLA, beta-2M, we have positive scores. We have different integrins that contain 2 different subunits. For example, LFA1, it contains both CD11A and CD18, and they are also showing positive scores. You can also start to see more complex structures. For example, here you see multiple proteins that are all colocalizing with each other. And these are then examples of these multi-protein domains. For example, here you can see a tetraspanin-rich domain. Tetraspanins are well known to localize with each other. You also have lipid raft-associated molecules showing high degrees of colocalization, etc. So let's focus then a little bit on the CAR receptor and what interaction partners the CD19 CAR receptor has. So here is the colocalization network of the CAR receptor. So these are all the proteins that the CAR receptor is colocalizing with. And you can see that here we get different adhesion molecules, for example, the different ICAMs, CD43, 44. We also get metalloproteases, ADAM10 or CD156C, which is another name. Different co-stimulatory molecules, so co-receptor CD8, and the SLAM F6. And interestingly enough, we can also see colocalization with the TCR. And you can also start to look at what proteins are actually not colocalizing and segregating from your CAR receptor. So some examples here were different lipid raft molecules and these 2 tetraspanins. And this is really interesting because the TCR has also been reported to not localize with lipid rafts at resting state, but during activation, it actually reorganizes into the lipid raft domain. On the other hand, the TCR is also in steady state colocalizing with these tetraspanins, and tetraspanins are really important for regulating organization, and they also play a role in synapse formation, etc. Whereas the CAR receptor is not at all colocalizing with these tetraspanins, possibly explaining differences in the CAR synapse where you don't really get these traditional ring structures, but more puncti, etc. So it could be a kind of differential regulation depending on which proteins it's interacting with. So in the next step of this project, we were interested in looking at what happens to the cells during functional responses. So we co-cultured our CAR T cells with the CD19 positive tumor cell line, Raji cells. And then we took samples either of T cells cultured alone, cells that were co-cultured for 4 hours or for 24 hours, and then we ran the Proximity Network Assay. So here you can see some protein abundance data and how protein abundance levels changed as compared to the T cells alone. So there was a lot of markers that were upregulated already at 4 hours, but maybe particularly at 24 hours. For example, co-stimulatory, some checkpoint molecules like TIM3 and PD1, etc. You can also see a very drastic down regulation of the CAR receptor. And this was seen actually already at 4 hours. And this can be either masking of the epitope because of CD19 binding. It could be internalization or it could be shedding. Quite interestingly, we also see actually loss of the TCR and different TCR-associated molecules like CD28 and 2B4. And I mean, these cells shouldn't really have any antigen specificity, but possibly this is a result of the fact that the CAR and the TCR is organized in similar domains and thereby affected at the same time. Something that we noticed when we looked at the protein abundance was that we also had upregulation of ICAM1. So here to the left, you can see a UMAP displaying the individual cells from the different samples in different colors here. So if we focus a little bit on this dark green, which is the 24-hour co-culture cells, and we focus here on the CD8 cells, you can see that ICAM1 seemed to be quite upregulated on these T cells. So we decided to visualize ICAM1 on these cells. So here I'm showing you a single T cell where we're plotting 3 different markers. And when we looked at ICAM1, we could see that it was showing this very patchy phenotype. So part of the protein was kind of evenly distributed on the cell surface. And then the cell had this very intense puncta, very high ICAM1 expression. We then decided to visualize some B cell markers, for example, CD40 and CD21 and 22. And we could see that these proteins were also co-localizing to a high degree with this ICAM1 rich kind of puncta. And on the other hand, we kind of saw loss or kind of absence of CD8 or other T cell related markers in these ICAM1 rich domains. So this really told us that these cells were in fact not upregulating ICAM1, but instead they were acquiring B cell membrane, so containing many different B cell markers. And this is a really kind of important feature that since you have this spatial information, you will be able to tell if you have true upregulation or if you have trogocytosis, even in the case where you have markers that are actually expressed by both of the cell lines. So we could determine that we had trogocytosis in this co-culture system. So we went ahead and we tried to quantify the degree of trogocytosis. So here you have 4 different T cells with different amount of tumor patches. So one T cell that has 0 patches, one with 1 patch, 2, 3, etc. And then what we did is that we divided the CAR T cells into 3 different groups. So either those that we called non-activated, that were not expressing CD25 or PD-1, those that were activated expressing CD25, and those that were late activation or exhaustion expressing CD25 and PD-1. And of course, in the sample with T cells alone, we didn't see any tumor patches since we don't really have any tumor cells there. But already after 4 hours, we can start to detect trogocytosis in these cells. And particularly after 24 hours, you see high degrees of trogocytosis with many of the cells having a multitude of tumor patches on their surface. And this was particularly prevalent in the late activated cells, so or exhausted cells that were having high degrees of CD25 and PD-1. And this is interesting because it's been shown before that trogocytosis can actually affect exhaustion of the cells, and that high degrees of trogocytosis are not really beneficial. Another thing that you can get information about in this assay is conjugate formation. So here's an example of a tumor tumor CAR T cell conjugate, where you can see part of this conjugate expressing T cell markers, for example CD3 and CD5, and the other part expressing B cell markers like CD22 and CD40. So since you have this spatial information, you can really visually inspect and understand if you have a cell that has high degrees of trogocytosis or if it is actually a doublet of a cell. And you can start to quantify which kind of cells are forming conjugates, how many conjugates do you have, and what kind of phenotype do these cells have. Okay, so to summarize this app note, CAR T cell phenotyping, we have shown you that you can use this FMC63-based detection to identify CAR T cells. In this case, we're doing in vitro or identification of in vitro generated CAR T cells. We have also had customers that have actually detected CAR T cells from patient samples, so after a time after injection. This FMC63-based CAR, we're kind of showing you a little bit of the interaction partners and its organization. So some things that we discovered was that this particular CAR receptor, it co-localizes with the TCR, and it shares several co-localization partners with the TCR, like CD8, CD5, and different ICAMs. And both of these proteins are also segregating from lipid rafts. However, unlike the CAR, the TCR actually co-localizes with tetraspadin CD81 and CD53. And of course, it's interesting to understand how different CAR receptors are affected by their kind of receptor design, and how this can change which co-localization partners they have, and how this can actually change their signaling capacity and synapse formation capability, etc. This assay also allows quantification of the synapse formation and to measure the degree of conjugates you get, as well as doing analysis on trogocytosis. And what is really key here is that you can also do this kind of measurement for proteins that are actually expressed by both cell types. Since you have this spatial information, you will get to see if the protein appears to be in patches with other T cell markers or other B cell markers, etc. So in general, the proximity network assay can be very efficiently applied to do deep phenotyping and functional assessment of CAR T cells. And ultimately, we believe that this can really aid in the identification of new biomarkers and help in the engineering of new improved CAR T cell therapies. So here you can again see a 3D layout of one of these CAR T cells that is having a tumor patch on its surface. So just as a final slide, I would like to just summarize the product that we provide. So as I mentioned, this is a sample prep kit. It doesn't require you to buy any instrumentation. You can use this kit in any lab, basically. It's just easy pipetting and different washing steps. And then finally, you will generate a sequencing ready library, and this library can be sequenced on any Illumina platform. So you can do it on your own sequencer, on a collaborator, or even on a facility platform. It allows you to analyze 155 protein markers, and in each kit you get 8 different reactions, and you can analyze up to 1,000 cells per sample or per reaction. When it comes to the analysis, I've shown you some example of what the data can look like, but we also have loads of other examples on our webpage, and feel free to reach out to us if you have any particular application examples that you're interested in. We also provide tutorials to help get you started with analysis, and it is a free software, so you don't need to buy any specific analysis pipeline. Okay, I think with that, I would like to end this webinar, and let's look at some questions.
Thank you so much, Hannah. Let's give them some time to ask questions. By the time, I can just ask a very general question about the resolution of our technology and the product. How is it in comparison to other technologies?
So it's a little bit difficult to assess exact resolution, since this is not really a microscopy technique. We don't take images of the cells, but rather we kind of detect unique molecules on the cell surface, and we detect them if they are close to each other, so to say. In a general experiment, we get around 50,000 molecules per cell. And when we have done calculations on T cells and their surface area, that gives us a resolution of about 50 nanometers between the individual proteins. If you have cells that express very high densities of proteins, for example, different cell lines, like Raji cells, you can get even up to 150,000 molecules per cell. These cells are, of course, bigger as well. But you really get high resolution spatial information from one of these assays.
That's great to know. Another question that I had was how easy was to do these co-culture experiments, and then later on perform a proximity network assay on it.
So in this particular experiments, we used an effector to target ratio of 1 to 1. So we had 1 CAR T cell to 1 tumor cell. We had quite efficient killing. So since we had so many CAR T cells, we actually did not purify them before the analysis. So we just took all the cells and we ran the proximity network assay. And then informatically, you can of course detect which are your tumor cells and which are your CARs or your non-transduced T cells. If you're doing experiments where you have, for example, tumor infiltrating cells, or where you have very low amounts of CAR T cells as compared to tumor cells, you can do a pre-enrichment. So you can select your T cells using flow cytometry or like FACS sorting or bead sorting to enrich that population, and then you can run the assay itself. So this depends a little bit on your experimental design, I would say.
All right. And I have one last question. So what do we offer? We give them a spike-in protocol, and can you talk about that a little bit?
For the CAR detection, you mean?
Experiments, yes.
So the product that we sell is as I mentioned, 155-plex panel. This panel contains markers for PBMC analysis. So it has markers for all the major cell types in PBMC: B cells, T cells, memory, naive, etc. We have recently done a deep annotation of PBMC, and we can detect up to 35 different immune cell types. I'm happy to show you a list of exactly which markers are expressed in different cell types. And then in addition to this, we can provide this CAR T cell detection, which is recognized in the FMC63 based CAR. And this allows you to analyze CD19 specific CAR T cells, basically.
Yes. I don't see any new questions.
And if any questions pop up afterwards, feel free to email us at hello@pixelgen.com or to my private email as well.
Yes. And we would also like to say that if you're interested in having a seminar being given in your institute or your university, please contact us at hello@pixelgen.com, or you can visit our shop page where you can see the business development managers in your region. You can contact them and have a chat with them about how this can be executed. All right, then. Thank you so much, Hannah. I guess we wrap it up here. Thank you, everyone, for joining, and we will see you at our next webinar. Thank you.
Thanks a lot. Bye-bye.
Bye.